This protocol enables the detection of tumor-associated mutations and currently complements invasive surgical procedures. Another strength is the ability to use this protocol to monitor tumor mutation load over time. The technique enables tumor genotyping without requiring actual surgical tissue biopsy, which is particularly helpful for tumors in challenging locations like the brainstem with restricted tissue access.
In the absence of repeat biopsy, longitudinal molecular data isn't available to complement MRI. Digital PCR monitoring facilitates a molecular-based supplement to diagnosis, enabling a more informed treatment decision-making process.
Similar methods can be used to detect the expression of genes in particular system with conventional fluorophore and quencher-linked probes, but with a higher sensitivity than with a quantitative PCR.
Before beginning the droplet digital PCR procedure, clean the bench space and equipment with 10% bleach and 70% ethanol, and gently vortex and briefly centrifuge all of the reagents except the droplet stabilizing oil. Confirm that the compressed nitrogen gas cylinder is attached to the droplet generating instrument and that the cylinder tank is set at 90 pounds per square inch. Add 38 microliters of digital PCR reaction mixture directly into the bottom of each well of an 8-well PCR tube strip, removing any bubbles with a clean pipette tip. Add 12 microliters of pre-amplified DNA sample in triplicate to the appropriate wells of the tube strip. Gently pipette the full volume 10 times to mix the reaction mixture with the DNA sample, and pipette the full volume from each well into the corresponding A through H channels of a droplet-generating instrument chip.